1. What information needs to be included when labeling microbiology experiments like agar plates or test tubes? Where does it go? Label includes your name, date, type of sample, and type of media. This label goes on the bottom of the plate, not the lid, and on the glass part or tape for tubes. 2. At the end of lab, where do you put the following items? Used Petri plates, biohazard bin, used test tubes, designated test tube disposal rack. Cultures, back to culture rack or autoclave. Gloves, biohazard bin. How do you place an agar plate in the incubator? Lid side up or down? Lid side down to prevent condensation from dripping onto your culture. 4. What is the scientific method? A logical process for asking questions and answering them through experiments and observations. 5. What are the steps of the scientific method in order? 1. Observation. 2. Question. 3. Hypothesis. 4. Experiment. 6. Conclusion. 6. What's the difference between dependent, independent, and control variables? Independent variable, what you change. Dependent variable, what you measure. Control variables, what you keep the same. 7. What are selective, general purpose, and enriched media? Selective media allows only some microbes to grow. Example, McConkey agar, general purpose media, grows a variety. Example, TSAya, enriched nutrients for picky microbes. Example, blood agar. 8. What are fastidious microorganisms? What type of media do they need? Fastidious microbes have special nutrient requirements and need enriched media, like blood agar. Quick review, Topic 1, Ubiquity and Scientific Method. Always label the bottom of plates with name, date, sample, media. Dispose of Petri dishes and gloves in the biohazard bin. Scientific method, observation, hypothesis, experiment, result, conclusion. Selective media supports only certain microbes. Fastidious bacteria grow only on enriched media like blood agar. Topic 2, Intro to Microscopy. 1. Important definitions. Magnification, enlarges the image. Resolution, ability to distinguish two close points, affected by wavelength of light and numerical aperture. Shorter wavelengths, higher NA, equals better resolution. Numerical aperture, NA, light gathering ability of the lens, increased by using better lenses or immersion oil. Contrast, difference between the object and background, improved by staining or adjusting light. Refraction, bending of light between different materials. What is the function of the objective lens? It magnifies the specimen and forms the initial image. 3. Objective lens types and magnification. Scanning, 4x, low dry, 10x, high dry, 40x, oil immersion, 100x 4. Function and magnification of ocular lens. The eyepiece, always 10x, magnifies the image from the objective lens. 5. What is total magnification? Multiply objective lens, ocular lens. Example, 40x objective times 10x ocular equals 400x. 6. Know the function and location of each microscope part. Illuminator, light source knob, adjusts brightness. Condenser, focuses light onto the slide. Iris diaphragm, controls the amount of light. Stage and clips, holds slide, adjustable. Nosepiece, rotates objective lenses. Coarse focus knob for large adjustments. Fine focus knob for sharp, small adjustments. Ocular lenses, eyepieces that magnify. 7. What should you do when moving to higher power? Center the object. Focus before switching. Only use fine focus under high power. This prevents crashing the lens and losing the image. 8. Which objective do you start with first? Always start with the scanning lens, 4x. 9. Which objective lens requires immersion oil? The 100x oil immersion lens, 10. What are the benefits of oil immersion? Improves resolution by reducing light refraction. 11. How does oil improve resolution? It allows more light to pass through the lens directly by reducing light scattering. 12. What's the relationship between magnification and field of view? As magnification increases, field of view gets smaller. 13. Why center the object before switching lenses? Because higher magnifications zoom in. If the object's not centered, you might lose it. 14. How do you estimate size of specimens? Use this formula, specimen size equals FOV diameter of specimens across view 15. What are the advantages of a wet mount? Lets you observe live organisms, their movement, and natural shape. 16. How can you tell if it's a eukaryotic cell? Prokaryotic cell or virus. Eukaryotic cells, larger visible nucleus. Prokaryotes, smaller, no nucleus. Viruses, extremely small, often not visible under light microscope. Quick review. Topic 2, microscopy. Start with the scanning lens 4x. Use oil immersion 100x for max detail. Resolution improves with oil and short wavelengths. Magnification goes up, field of view goes down. Wet mounts are good for viewing live cells. Eukaryotes have a nucleus, prokaryotes don't. Topic 3, simple and negative staining. 1. What are the main bacterial morphologies? Coccus, spherical, bacillus, rod-shaped, spiral, vibrio, comma-shaped. 2. What are common bacterial arrangements? Diplo, pairs, strepto, chains, tetrads, groups of 4, cubes of 8. 3. Why do we stain microbes? Because most microbes are colorless and hard to see without contrast. 4. What is a smear and how do you make one? A thin layer of microbes spread on a slide, air-dried, then heat-fixed, except for negative stains. 5. Why heat-fix a smear? To kill bacteria and make them stick to the slide. 6. Do we heat-fix for negative staining? No, we don't heat-fix because we want to preserve the natural shape and size. 7. Advantages of negative staining? Better for seeing capsules. Doesn't distort shape, useful for delicate cells. 8. What info do you get from simple and negative staining? Simple stain, cell shape, size, and arrangement. Negative stain, cell outline and external features like capsules. 9. What's the difference between basic and acidic dyes? Basic dye, positively charged, sticks to negatively charged cell wall. Acidic dye, negatively charged, stains the background. 10. When would you use each? Use basic dye for simple staining, colors the cells. Use acidic dye for negative staining, colors the background. Which stains are used for each method? Simple stain, basic dye, negative stain, acidic dye. 12. Examples of basic dyes used in lab: crystal violet, safranin, methylene blue. 13. Name of the acidic stain we used: negrosin. Quick review. Topic 3, staining. Coccus equals round, bacillus equals rod, spirillum equals spiral. Use basic dyes for simple stains, acidic dyes for negative stains. Do not heat fix negative stains. Negative stains show capsules and shape clearly. Negrosin is the acidic dye we used. Let me know if you'd like the audio file version or if you'd like flashcards or quiz